We study C. elegans, and in particular we study cilia in C. elegans. And cilia are extensions of some of the cells in C. elegans, of some of the neurons in C. elegans. In fact, they act as sort of antenna connecting the animal with the outside world. And in particular measuring, sensing, or maybe tasting is the better word, chemical compounds in the environment. And we try to understand how this sensory mechanism works in living worms. And this works with these cilia, these special organelles. And we really need fancy microscopes to look at them and to see how they work. Overall the goal of my research is to understand how life processes, how they work and what their physical origins are. And there is, in these cilia, in these worms, there is several of these processes that we are really excited about. How do certain proteins end up at a certain location in a combination of active transport driven by motors, intermolecular transport, but also diffusion, random motion. We try to understand how that works. But also we try to understand how this actual sensing works, how the interaction of a single molecule with a single receptor, how that translates to an avalanche of effects, neuronal activity, and then ultimately to changes in behavior. That's what is so fascinating. I had been studying them for a long time in vitro conditions, so isolated proteins working together under the microscope and looking what's happening. And that's great, fascinating, you can learn a lot. But biology in the cell is so much more complex, and that's why we switched to an in vivo system. And C. elegans, I was introduced to John Scully, a collaborator, to this system, and that really opened my eyes. It's so much nicer to really look into this in vivo system and to really try to understand life in its full complexity. And C. elegans is really super unique in the sense that you can really go from the single molecule level to really the behavior of the whole animal. And I don't think that's possible with any other model system. Imaging, and to be more precise, fluorescence microscopy is really the cornerstone of our research. You wouldn't be able to see anything without a microscope. Things are too small, and we need fluorescence for the specificity. We are interested in single molecule behavior, and cells contain thousands, millions of proteins, but we really need to make sure that only one of them is labeled and visible, and we need the sensitivity and the specificity, and there's only fluorescence microscopy can bring that to us. We study living systems, and for that it's really important, first of all, as always, to be sensitive, very high sensitivity, very high specificity, suppress background very well. But another crucial thing is that imaging needs to be fast. And maybe the most important one is that imaging needs to be gentle. There are several reasons for that. Photobleaching, that is, the fluorescence signal decreases over time. That makes your images fade. That's not good. But more importantly, too much light can be toxic to the animal, and then we're really changing all sort of processes in the animal and while we're studying it, and that's not what we want. So the NL5 has enabled us to measure in different parts of the animal where we so far could not measure properly. Because the animal is relatively thick in certain places, and that makes imaging difficult. The NL5 really helps suppressing the background. Furthermore, what we really notice is the ease of use and the relative simplicity as a user of this instrument that really allows you to play a lot with the settings and find the things that work. And what really stands out is that it is very gentle to the specimen. We can really measure very, very long times and get very long sequences of images, and that really benefits our research.
So in our lab we look at these chemosensory cilia, and we've looked at how they function, specifically their transport. And in my research I look more into how they actually sense. So to do this we put the worm inside a very small microscopic chip, and then we can very precisely expose the worms to different kinds of chemicals, and then we can study with our NL5 system how the cilia and the whole nervous system reacts to these types of stimuli. So what is important for my research is that, especially when I look at these cilia, is that we need optical sectioning. And in the tail of the worm there's only 1 or 2 cilia, but in the head of the worm there are many cilia that are combined. And of course this is in 3D, so we need this optical sectioning to really get a better contrast on the processes that we see. And we're looking at processes of tiny motor proteins with very low signal. So that's why we need also the high sensitivity of this system.